Hello, student. My name is Laura Antonez, and today I am going to guide you through the Utah Genetics Gel Electrophoresis virtual lab that your teacher has assigned to you. Let's read the directions together. This lab will walk you step by step through a virtual gel electrophoresis. Follow the directions in the yellow box at the bottom of the screen and use the forward arrow on the bottom right corner on the screen to move to the next step of the lab. There are a few steps where you will need to move something from one place to another or pour liquid out of a container. To complete this step, click and drag the item you want to move and it should move it for you. When you get to the end of step number 5 of the lab, paste a screenshot of your completed gel electrophoresis and base pair estimates on the next slide and answer the 2 questions on that slide. All right, so when we go to the next slide of your assignment, we are going to be able to see that you must paste what they're asking you to paste right in here. And that's a screenshot of your complete gel electrophoresis right here, adding your estimates of the base pair size for each of the 3 labeled DNA molecules in the spaces to the left of the gel. All right, after that, we will be answering together these 2 questions. Let's go to the lab and let's start. All right, here we are, gel electrophoresis. Remember, the word electro means electricity, and the word phoresis means transport. All right, so you are transporting electricity through the liquid. That's what electrophoresis means. Right, have you ever wondered how scientists work with tiny molecules that cannot see? Here's your chance to try it yourself. Sort and measure DNA strands by running your own gel electrophoresis experiment. This is going to be really cool. All right, this is you in a lab and you're holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? We're going to click forward, all right? And we can see the strands of DNA in here, and they are either short, they are medium, or they are large. Look at this tube in here. But DNA strands are molecules so tiny that you can see them even not under most microscopes. Is there a way to start to sort and measure the DNA strands in your tube, even though you can see or touch them? Yes, there is. It's called electrophoresis. Very good. Excellent. Let's do it. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How do they do it? Let's find out. Okay, let's turn on the machine. Just like plug the end of the black wire into the black plug to generate a negative charge, and same thing with the red one to generate a positive charge. And now let's turn on the machine. These bubbles are your proof the current is running. Okay, so let's finish your work. First, we are going to paste the screenshot that we took from our lab in here. Okay, let's make it a little bit smaller. And finally, we are going to answer to the two questions to complete this lab. How were you able to estimate the base pair sizes of the three labeled DNA pieces in your gel electrophoresis? All right, think about it. Take a minute to answer this question. Maybe you want to say something like you were able to estimate the base pair sizes of the three labeled DNA pieces by comparing them with the DNA size standard chart. Excellent. And to finish, what is one new thing that you learned about the gel electrophoresis from this lab? I'm going to leave that answer for you. It was a pleasure to be with you today and see you in the next video. Bye-bye.